Come learn about the biological macromolecules with me as I make my breakfast. When people talk about macros in terms of diets, they are referencing the main biological macromolecules. Every ingredient on this plate was once part of a living organism that needed these molecules to survive, grow, or store energy. Today, we're going to break down the 4 major macromolecules of life using my breakfast. We'll talk about what's in the food and why it's there. So, on the docket, we have carbohydrates, lipids, proteins, and nucleic acids. First up, let's explore the carbohydrates through the base of my breakfast, some toast. Okay, so checking out the nutrition facts on a basic loaf of whole wheat bread from my local grocery store, we can see that a slice of bread has about 18 grams of carbohydrates. But you'll also notice that nestled under these categories of carbs, there's 2 distinct subgroups here: sugars and dietary fiber. Let's learn about the difference. The fundamental building block, or monomer, of carbohydrates is the monosaccharide, sometimes referred to as simple sugars. A prime example is glucose, a simple sugar that serves as a primary energy source for cells. All carbohydrates are composed of carbon, hydrogen, and oxygen, typically following a really strict 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. In the case of glucose, this results in the molecular formula of C6H12O6. This specific ratio is a hallmark of hydrated carbons, where every carbon atom is essentially associated with the components of a water molecule, providing a highly efficient structure for quick energy release. When these individual monomers are linked together through dehydration synthesis, they form polymers, known as polysaccharides. This transition from monomer to polymer is how organisms store those sugar units for later use without affecting the cell's osmotic pressure. For example, plants link glucose monomers into amylose, which is a linear, unbranching chain. Amylose is a key component of starch, along with amylopectin, acting as the plant's primary energy warehouse. Many plants, like potatoes, are packed with starch, which makes sense if that little potato is going to grow and grow into a whole new plant. In contrast, animals and fungi utilize glycogen for energy storage. While glycogen is also a polymer of glucose, it's much more highly branched than amylose. This branching allows enzymes to break down the polymer into individual glucose monomers really rapidly when an animal needs a sudden burst of energy. In this context, it makes tons of sense that your muscles would store glycogen. Your muscles are always moving and in constant need of energy from glucose. So why are these carbohydrates even in my slice of toast? Well, remember that toast is made of wheat, which is a plant. Wheat plants produce glucose through photosynthesis, but instead of using it all immediately, they can link those sugar monomers together into large polysaccharides, like starch, to serve as long-term energy storage. The starch is concentrated heavily within the endosperm of the wheat seed. From the plant's perspective, this isn't really just a food source for humans, it's a biological starter kit for the wheat embryo. So when that seed eventually germinates, it will break down those stored carbohydrates to fuel its initial growth until it can start to grow leaves and begin making its own energy from the sun via photosynthesis. Now, a question that is often asked is carbs bad for you? The short answer is no, that's way too big of a generalization. Personally, I think that there's 2 things that need to be considered. First, chemical structure relating to how it's processed in your body, and then also context. When you consume something like toast or a banana, your body handles the simple and complex components quite differently based on their molecular architecture. So those simple sugars, like monosaccharides or even disaccharides, like the sucrose found in a can of soda, these are fast cash of the metabolic world. Because they are really small, they are absorbed readily through the small intestine, leading to a quick spike in blood glucose that can make you feel shaky and then later crash. In contrast, complex carbohydrates, like the starch in a banana, consist of long, intricate chains of glucose. They're polymers. So your digestive enzymes must work like a construction crew to systematically break down the chemical bonds between each monomer. This process takes more time, resulting in a slower, more sustained release of energy into the bloodstream, which prevents that crash that you sometimes get from the high sugar foods. Also, like I said, context is really important. So if I'm about to embark on a 30-mile bike ride, I usually keep a bag of high sugar gummy bears literally attached to my handlebars because I need and will be using that quick energy source. However, if I were instead sitting down and working at my computer, that's going to influence how those gummy bears are metabolized in my body. Instead of being immediately useful, my body will probably store excess sugar for later, sometimes in the form of fats. Though I must admit I am oversimplifying the process. Then finally, we have the category of fiber. Fiber represents a unique category of complex carbohydrates, and actually humans just put them into this category because we can't digest them or break them apart. That's the only difference. Like the starch, it's a polysaccharide, but fiber, like cellulose found in wheat bran, is held together by specific bonds that our digestive enzymes are unable to break. So, because we can't clip these monomers apart, fiber passes through the digestive tract largely intact. Instead of providing calories to us, it provides the physical bulk needed to keep the digestive system moving and serves as a vital food source for those beneficial bacteria in your gut microbiome. That's why something like whole grain toast tends to be more satisfying than a white bread. White breads usually have more simple sugars. And the whole wheat bread is going to contain those structural components of the plant that your body has to work harder to process. And like I mentioned, it feeds your super important microbial symbionts. Let's jump into the world of lipids by shifting our attention to the next part of my breakfast, the avocado. Avocados are unique because they have a high proportion of triglycerols, aka fats, rather than sugars. While the seed inside an avocado fruit contains the instructions and fuel to grow a new tree, the avocado tree actually invests a massive amount of energy into the fleshy part of the fruit in order to attract large animals. Historically, this would have been megafauna, like giant ground sloths, which no longer exist today and you should definitely look up. These animals would eat the fruit whole and then later deposit the large seed elsewhere, aka poop it out, so that the seed is moved to a new location and conveniently packaged in a pile of fertilizer. So the tree packs triglycerides, aka fats, into that outer fleshy green part of the fruit to entice seed dispersers. Triglycerides represent highly concentrated energy storage, containing more than double the energy per gram than carbohydrates. Again, double the energy per gram than carbohydrates. That's one of the important reasons that we store fat on our bodies. But you'll notice that the nutrition labels even break down the fat category further, which we'll discuss soon. First, know that there are actually more to the lipids category than just the fats or triglycerols. We also have the phospholipids and steroids. The triglycerides we already learned a little bit about. Structurally, they consist of a glycerol backbone attached to 3 fatty acid tails, like you can see on this image all the way to the left. While the triglycerides store energy, phospholipids are a very important structural component of every cell. So every single cell in that avocado is encased in a plasma membrane made of a phospholipid bilayer. These molecules are amphipathic, meaning they have a phosphate head that loves water and is hydrophilic, and 2 fatty acid tails that hate water and are considered hydrophobic. This unique structure allows them to form a protective barrier that keeps the inside of the cell separate from the outside. Without these specific lipids, the avocado wouldn't have any structure to it at all. It would just be a puddle of biological molecules rather than a firm, sliceable fruit. Finally, we have the steroids, the third category. They're characterized by a structure of 4 fused carbon rings rather than these long hydrocarbon tails. While we often think of cholesterol in humans, plants, like avocados, contain phytosterols. These molecules sit within the phospholipid bilayer of the avocado cell membranes, acting as stabilizers to keep the membrane from becoming too fluid or too rigid as temperatures change. They are essential for maintaining the integrity of the plant cells, ensuring the avocado stays fresh and functional while it hangs on the branch. In humans, cholesterol is kind of like a double agent. It is both an important structural component of cell membranes, just like those phytosterols, and it's also the starter molecule for all other steroids or lipid-based signaling molecules in the human body. For an example, testosterone is a steroid, and to make it, our bodies take cholesterol and modify it. So you can see that the lipids are really diverse in terms of structures and functions. However, one way to recognize a lipid is to see this high proportion of hydrogens and carbons, which generally make these molecules hydrophobic or water-fearing, of course, with the exception of those phospholipids, which are amphipathic. Interestingly, the chemical structure of the fatty acids in a fat molecule determine whether they are solid or liquid and how they impact your health. So, saturated fats have straight hydrocarbon chains in their fatty acid tails because every carbon is saturated with hydrogen atoms, allowing them to pack tightly together and remain solid at room temperature. Unsaturated fats, like heart healthy oils in the avocado, contain one or more double bonds. In their cis configuration, meaning that the double bond creates an orientation where the remaining hydrogens are on the same side of the tail, there's a kink or a bend in the chain that's created. And this prevents that tight packaging of the fats, keeping them liquid at room temperature. So the olive oil that I use to coat the pan to make my eggs contains a lot of unsaturated fats, and these are considered healthy. However, trans fats, which are unsaturated fats, are a really dangerous middle ground. They're often created through industrial hydrogenation to make vegetable oils more stable. You've probably heard of hydrogenated oils. This process straightens out the kinked tails, making the fat behave more like a saturated fat structurally, but with a configuration your body just does not process well. So, trans fats are significantly worse for your health because they not only raise bad LDL cholesterol, but also actively lower this good HDL cholesterol, drastically increasing the risk of heart disease compared to their natural counterparts. Not to worry though, in 2015, strides were made to limit trans fats in foods. So the FDA made a landmark ruling that partially hydrogenated oils, the primary source of artificial trans fats, were no longer generally recognized as safe for use in human food. This was a massive shift, and it moved trans fats from being a regular ingredient to essentially an unapproved food additive. Natural trans fats actually do exist in small amounts in meat and dairy, like the eggs in my breakfast, because they are produced by bacteria in the guts of ruminant animals. However, this is really in small quantities and it's nothing to really be sweating over in moderation. So, now to the final ingredient of my breakfast, the eggs. Eggs are famous for having protein in them, specifically albumin. Let's look at what a protein is structurally. So, starting with the monomer, aka the single building block of a protein, we have an amino acid. It contains a central carbon that's going to be attached on all four sides because carbon can make four covalent bonds. So, first, we see that it's attached to an amino group, a nitrogen and two hydrogens. That central carbon is also attached to a carboxyl group, a carbon double bonded to an oxygen, and also bonded to an oxygen and a hydrogen. It's additionally bonded to one singular hydrogen, and then the R group or side chain. Note that R here is not an element, it's a placeholder because there are many different amino acids and they have different identities because of those different side chains. The backbone of the amino acid is everything but the side chain. So all the nitrogen, hydrogen, carbon, and oxygen that you see here, this is the same for every single amino acid. So how do we build a protein? Well, we take our amino acid monomers and we linked them together to form polypeptides, which is a polymer. When two individual building blocks like these two amino acids here come together to bond, they can't just stick to each other as they are. We've got to clear up some space. So one amino acid is going to give up a hydrogen atom, and then the other one is going to give up a hydroxyl group, or an OH, oxygen, hydrogen. When these two pieces leave their respective molecules, they find each other and combine to form a single molecule of water, H2O. Because a water molecule is being removed or lost from the original pair, we call this a dehydration reaction, and specifically a dehydration synthesis reaction because it helps us build macromolecules. So once that water is out of the way, the two building blocks have these open hands, aka available chemical bonds, to reach out and grab onto each other. This creates a new strong link called a peptide bond in the case of the protein, and note that it's a covalent bond. So there's 20 different amino acids in total that the human body uses as the universal building blocks for making proteins. While they all have the same general structure, remember that the R group or variable side chain is different. So in this figure on the right, you can see that the different amino acids are categorized based on chemical properties of those side chains, such as if they are hydrophilic or hydrophobic. Also, as a fun side note, a lot of protein powders that you find at the grocery store have an amino acid breakdown on their labels, and this is exactly what they're referring to. For an example, if you were to buy a collagen powder, collagen is a protein, and the powder should probably contain all of the amino acids that are used to build collagen. So, proteins have a very complicated structure, and it's divided into 4 parts. The primary structure is the unique linear sequence of amino acids linked together by those peptide bonds. You can think of this as like the spelling of every protein. Even changing a single amino acid in this chain can actually completely alter the protein's final function. This sequence is also determined directly by the instructions found in your DNA. Secondary structure consists of local patterns that form when the polypeptide backbone folds or coils due to hydrogen bonding between the oxygens and hydrogen atoms of the amino acid backbone. The 2 most common shapes that we see are the alpha helix, like a delicate coil, and the beta pleated sheet, which is a series of folds. These structures are independent of the amino acid side chains and provide the initial scaffolding for the protein's shape. The tertiary structure is the overall 3-dimensional shape of a single polypeptide, which is determined by interactions between the R groups, or those side chains of the amino acid. These interactions include hydrophobic hiding from water, ionic bonds, disulfide bridges, etc. At this stage, the protein folds into its final functional glob or fiber. For many proteins, this is the final step where they become biologically active. However, there can be a quaternary structure. The quaternary structure only occurs in proteins that are made up of more than one polypeptide chain working together as a single unit. These multiple subunits aggregate into a larger complex held together by the same types of interactions found in the tertiary structure. A classic example is hemoglobin in the blood, or certain complex enzymes found in egg whites, which require multiple pieces to snap together like a puzzle to function properly. In biology, we often see that structure is directly related to function. So, a protein's physical shape is precisely engineered for its specific job. If that shape changes, the protein can no longer do its work, much like a bent key failing to fit into a lock. So, collagen is an ultimate example of this. Its job is to provide strength and stretch and resistance to your skin, tendons, and bones. To do this, its structure mimics a heavy-duty rope. 3 long polypeptide chains are wound tightly around one another into this sort of triple helix formation. So just as braiding individual threads into a rope makes it strong enough to hold an immense amount of weight, this triple wrapped arrangement allows collagen to withstand immense physical tension without snapping. Now, proteins are really diverse. They're often called workhorses of the cell because they perform a diverse array of tasks, maybe more so than any other macromolecule. Their versatility is a direct result of their complex 3-dimensional shapes, which allow them to act as enzymes that catalyze chemical reactions, structural supports, like the collagen in our tissues, transporters, like hemoglobin that carries oxygen through the blood, or other transport proteins on cell membranes. And then, even beyond physical structure, proteins function as hormones for long distance signaling in the body, antibodies that defend the body against pathogens and work with our immune system, and contractile fibers like actin and myosin that allow our muscles to move. They can even be storage proteins, like how we see them in eggs. So while carbohydrates and lipids really do primarily serve as fuel and energy storage, proteins are the active machinery, they're super diverse, and they physically carry out instructions of the cell's DNA. So why would an egg have protein in it? An egg is essentially a biological self-assembly kit, kind of like a seed. It contains everything that's needed to build a complex living organism from scratch. So while the yolk provides the energy, lots of lipids and protein, a high proportion of protein is primarily found in the egg white, or albumin. It serves as the structural and functional raw material for the developing chicken. These proteins are the tools and bricks that will be broken down into amino acids and then later reassembled to form the embryo's muscles, skin, beak, and essential enzymes. Because the egg is a closed system, it really must contain a diverse and complete set of protein building blocks to ensure that the chicken has every necessary component to go from a single cell to a fully functioning animal. All right, now hold on. We've made it through my whole breakfast, but there's still one group of macromolecule we haven't discussed: the nucleic acids. The food we eat comes from other living things. For an example, a piece of kale was once a leaf on a kale plant, and that leaf is made up of many plant cells. And remember, cells are the building block of all life. Within each cell of a kale leaf, there's a nucleus, and it holds the genetic code for that plant in the form of DNA. DNA is a type of nucleic acid, and it's the instruction manual for life. As a side note, while you can break down nucleic acids as an energy source, they are not the primary energy source. Instead, all the DNA we eat from a plant, we can break down and then actually use to build our own DNA when we make new cells. So you literally are what you eat. Let's start with the monomer, aka basic building block of the nucleic acid, and this is called the nucleotide. To understand its structure, you can think of it as a three-parter. They all consist of a 5-carbon sugar, a phosphate group, and a nitrogenous base. The sugar is either ribose in the case of RNA or ribonucleic acid, or deoxyribose sugar in the case of DNA, deoxyribonucleic acid. This is going to serve as our central hub. Attached to one side of this sugar is a phosphate group, which acts as a linker that allows nucleotides to join together. On the other side is the nitrogenous base, which is the part of the molecule that actually carries the genetic information. We'll learn about that later. For now, know that there are 5 different nitrogenous bases: cytosine, thymine, uracil, adenine, and guanine, often referred to with just a single letter: C, T, U, A, and G. Let's broadly look at the differences between DNA and RNA. So DNA and RNA are the 2 main types of nucleic acid. They share that same structure, a phosphate group, sugar, nitrogenous base, with many of those nucleotides linked together, but they also differ in a few ways. So an RNA is single-stranded, whereas DNA is made up of 2 nucleic acid strands. So it's called double-stranded, and it forms that ladder-like double helix. We mentioned that RNA has that ribose sugar, whereas DNA contains deoxyribose sugar. Hence their full names, ribonucleic acid and deoxyribonucleic acid. And finally, while both RNA and DNA use bases cytosine, guanine, and adenine, DNA uses thymine, whereas RNA utilizes uracil. Think of DNA as the instructions on how to make you. When nucleotide monomers are linked together through dehydration synthesis reactions, the sugar and phosphate form a backbone of the nucleic acid polymer, with the bases sticking out like teeth of a zipper. So the specific order of those bases, A, G, C, and T, they literally spell out instructions, like letters do when we write a paragraph in English. DNA serves as the massive how-to guide for your wheat and your toast, avocado plants and eggs, and then of course your own cells. So as you take a bite of your breakfast with toast, avocado, and eggs, remember that your body is about to perform a series of hydrolysis reactions, which is the opposite of the dehydration synthesis reactions. And instead of building, we are tearing those molecules apart. So we'll be breaking down the wheat's carbohydrates into simple glucose for quick energy. We'll be dismantling the egg's proteins into a pool of amino acids so that we can build our own proteins. We'll be using the lipids as an energy source, and then our cells are going to use the nucleic acids to tell our body how to resemble those recycled parts into our specific muscles, enzymes, and our own energy systems. So that breakfast was more than just a meal. It was a delivery of raw materials and building blocks required to maintain the living machinery that is you.